The synthesis of a polypeptide by a eukaryote accounts for two processes, transcription and translation. Transcription is the process of copying a segment of DNA into mRNA. The first step of this process occurs through RNA polymerase, which separates the two strands of DNA in the 5'3" direction. The promoter is the start sequence for polymerase, and the terminator is the end sequence where the polymerase finally detaches. The end result of all this is RNA. Translation is the next step in making protein. RNA takes the genetic code which is carried by mRNA and translates it into a sequence of amino acids. The mRNA is read in triplets called codons, and each codon codes for a specific amino acid. The tRNA then carries the amino acids, these building blocks of protein, to the ribosomes to form a polypeptide chain. Ribosomes are instructed to start and stop through start codons and stop codons. The effect of a deletion or insertion in one of the DNA nucleotides causes the amino acid to change completely. Both insertion and deletion are types of frameshift mutations. Insertion is the addition of a nucleotide, and deletion is the removal. As you can see, while the DNA is written with the addition of a nucleotide, the codons are completely different with the exception of the first two in the mutated strand. While transcribing this sequence to mRNA, the codons are different as well, and while writing them as amino acids, it is seen that they code differently also. Effective substitution in one of the nucleotides most likely changes only one amino acid in the sequence. For a silent mutation, there is no effect on the amino acid sequence. As you can see, A is substituted with a G and is transcribed to CUC, which still codes for leucine. For a missense mutation, depending on the location, the codon will code for a different amino acid. This is most common. Since A is substituted with a G, leucine becomes proline. For a nonsense mutation, the altered codon will become a stop codon. This results in a shorter, unfinished protein and a change in the base of DNA that stops the translation of RNA.